really great to join the second World CRISPR Day and to present our work. And just a quick overview of what we do in the research lab. And our research focus is to think about beyond the gene editing. And what we have done before is to apply synthetic biology and protein engineering to create novel variants of the CRISPR molecules harvested from nature that you heard from the previous talks, and give them new functions that doesn't normally exist in these natural systems. One example was the use of the Cas9, which by silencing the nucleus domains of the molecule, but still make use that as an RNA-guided DNA binding system for controllable genome regulation, known as CRISPR-I, CRISPR-A. And the same principle has been also applied to other systems. So the concept is we have a class of programmable molecules, Cas9, 12, 13, combined with guide RNAs, delivered to the cells, and depending on the nature of these molecules, either they bind to DNA or RNA, so we can use them for some functions beyond editing. So for example, in the CRISPR-I and CRISPR-A, one for inhibition, another for activation, we can use this Cas9, Cas12 system to, instead of cutting the DNA, recruit an effector to turn on or turn off the downstream gene regulation. In a similar manner, we can also recruit enzymatic domains to either methylate or acetylate the DNA or chromatin to change the chromatin status to rewrite the epigenetic memory. And as another example, we can also recruit the pieces of DNA to modulate how the DNA interact with each other and also localized differently in the nucleus as a 3D genome control. And also this tool can be repurposed for genome or RNA imaging by fusing them to dyes. And in the last talk that came to the CRISPR Day meeting, I presented how we repurposed a bacteria antiviral system, which is this CRISPR-Cas13, as a human antiviral system to fight SARS-CoV-2. So really, like for our interest is we like to think beyond the box, outside the box, to how we can use these tools for different purposes. Despite there are many, many exciting and interesting CRISPR applications for research, for screening, for cell engineering, there still remains a key challenge, which is how can we use these tools more effectively, safely, and programmably in vivo, in animals or in the human body. And if you look at the practical limitation of the CRISPR system, it's not very friendly for delivery. Really, like I'm using the adeno-associated virus, AAV, as an example. So the Cas system naturally come at the big size shown just for your reference. And barely, most CRISPR systems do not fit into a single AAV vector. And there are only a few examples that have been discovered before, like the Staphylococcus aureus Cas9, 3.2 kb, or a CG Cas9, 3.0 kb, can barely fit into a single AAV, yet the efficiency is not great. And also further expanding into other applications by fusing the Cas with effectors for activation, repression, base editing, prime editing, or epigenome editing, like what we really love to do, is there is no way to solve this problem. It's definitely not good for a single vector. The question we ask is, how can we apply synthetic biology and protein engineering to make this become a possibility? And that's promote us to work on a much smaller, highly compact miniature CRISPR, we termed Cas-Mini. Before we engineer Cas-Mini, we look at the naturally discovered CRISPRs. And among the Cas9 family, they are very large, generally larger than 1,000 base pair. Among the Cas12 family, there are some interesting subgroups, like Cas12F, also called Cas14, and their size is in general smaller than 1.6 kb. So when we compare the structure, to be honest, when we started this project working on engineering the Cas12F, and there was not a structure reported for this system. So we had to computationally predict it. And then by aligning it to other Cas9 and Cas12 the structures. Cas9 has 4.2 kb. It's a large protein with many domains. Compared to that, the Cas12F has a smaller structure and also is from a dimer And that's why even though at 1/3 of the size of the Cas9, because of this dimerization, it can do something that similarly as Cas9. That's why the system is super compact, but still working. But the first question is, okay, you get a system from bacteria. Does that work in mammalian cells? That's not guaranteed because these systems came from could be a simpler environment due to evolution. They may not have been prepared to work in a much larger genome, like our human genome, which is 1,000 times bigger than a bacterial genome. So the postdoc Xiaoshu worked on this question by engineering a mammalian version and by also mutating the Cas12f into a dead version. So it still binds but doesn't cut. That's a hope. And to our disappointment, the natural system did not work at all. And when we tried to activate a fluorescent GIP reporter in the human HAC93 cells, we saw 0% cells can be activated, suggesting the system completely failed in mammalian cells. Then we think about synthetic biology. How about engineering molecules? First is the guide RNA. And we look at the secondary structure of the guide RNA, and we did a few changes. One, to eliminate the transcriptional terminators. Another is to engineer at the interface between the CRISPR-RNA and the guide RNA, showing this blue box. And that slightly improves the performance. For example, compared to the version that has no effect at all, that we find one design shows 3.6-fold improvement. But while that's good, right? There's a trend, but it's far from being useful. Then we turn to protein engineering. By computational modeling, we choose different amino acids that are closer to the target DNA because we think that closer residues should have an important function for binding. And second is we mutate that to a positive charge residue due to this electron interaction, because DNA is negatively charged, and if it makes the protein more positive charged, we may increase the interaction with the target DNA. To our surprise, that greatly improved the performance of the gene activation on the fluorescent reporter. As you can see, some mutants even showed more than 100-fold improvement for the performance. And there's also a domain that seems to cluster together. And each residue within that domain shows a strong propensity for improvement. We also used repetitive engineering to iteratively engineer the protein with more mutations. So basically, we choose the good mutants from each round of mutation screening, and then we introduce more mutations by combining the beneficial mutants. Hopefully, these mutations will stack together. And that really leads to a gradually improved performance of the mutants, starting with some wild-type systems shown on the left that has almost no activity. And after each round of mutation, we find better and better variants. And as in the round 4, we find almost 1,000-fold improvement over the original system. So we also ask questions: how about that really compared to the original system on endogenous genes? We tested on about many genes, and shown here just 2 examples: the human interferon gamma and also the human beta globin gene. As you can see, the gray bar shows the original system performance, almost nothing, and the color bar shows how much we can activate this gene. And since these axes are on the log fold, so you can see there's a great improvement. And we also defined the requirements for the guide RNA to bind. And not every guide RNA will work. So to summarize this data, we test many genes and many guide RNAs. For each gene, we designed 10, 20 guide RNAs. And we found about 20% of the guide RNAs showed activation. And also, via this process, we better define the PAM sequence to be TTTR. So the next question is, how does this Cas-Mini system seems working well, but how does it compare to the other system? To do that, we compare to Cas12a, which is CPF1. As you can see, Cas-Mini has a much smaller size. We target design guide RNA, and for both systems target to the same region of the GIP reporter, because they can use the same PAM sequence. And the Cas-Mini showed even better activation on this reporter gene. When we compared across many endogenous genes, our conclusion is the Cas-Mini activator either showed similar performance or better performance on these genes, so which is very, very good. And the next question is the specificity, how many off targets? And then when we engineer a CRISPR system to be more effective, there is a natural question that also become less specific. To do that, we performed genome-wide RNA sequencing, and by comparing a targeting guide versus a non-targeting guide, and how does that affect the global-wide transcriptome expression. And the red colors show the Cas-mini influence on the endogenous genes, and the black dots shows the Cas12. From this data, you can see they are very comparable and not much off targets or no off targets. And while the on target gene is strongly activated. So we also use this engineered version to test on gene editing, since the system came as a nuclease system. We test on different genes and different loci and achieved about 10-15% of gene editing efficiency. We also compare different mutants and to see what's the editing window. And because people know Cas9 has a very narrow editing window, ±1 is a dominant editing. And for our system, we find a much broader editing window. More than 20 bits per DNA is deleted. So that would be a good news for knocking out a gene, because that would be a more complete disruption of the target gene. We also fuse the Cas-mini to the adenosine deaminase, which will mutate a single A to a single G. And we test different protein engineering constructs, and we found some constructs allow us to achieve a decent number of base editing at different genes. And also the editing window is very narrow, which is a good news because it doesn't affect A's outside of the editing window. And as you can see, the editing window is usually the third and fourth base pair. And we think the Cas-mini system really brings in new opportunities to work with this very limited delivery capacity of the many systems, including adeno-associated virus, and even for mRNA delivery, smaller payloads usually can be delivered more efficiently. And that's particularly pronounced when we work with different functions, repressor, activator, base editing, prime editing, because from our prediction, they all fall below the packaging capacity of the AAV. And so far in this work, we demonstrate the use of synthetic biology to turn a originally non-functional system, Cas12f or Cas14, into a completely working system, and also not only working, but working comparably well with other defined gold standard CRISPRs, and also demonstrate their use for different functions on the genome. As you see that this could be useful, very more friendly for different delivery approaches. With this, I'd like to thank the lab for contributing the work and providing the useful tools to the research community, and also enables potential use for the clinics. And in particularly the leading author, Xiaoshu Su, who's diligently engineered the protein and tolerate all the failures and until the system working well. And also the funding source. Thank you very much.